Hi everyone, and welcome to the 18th TI Centre seminar of the academic year. So my name's Adam Shepherd, and I'm a researcher here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and associated areas to discuss their work. And today I'm absolutely delighted to introduce Dr. Edwin Jong. Edwin has been fascinated by how humans and machines produce intelligent behaviour for over 3 decades. He studied at Delft University of Technology and received his PhD from the VUB AI lab in 2000. As a postdoctoral researcher at Professor Pollock's demo lab at Brandeis University, he started a research line in representation learning and open-ended self-improvement, which he continued in the Netherlands at Utrecht University. In 2004-2005, he co-founded Adapticon, among the first companies worldwide to apply LSTM in industry. And his current focus is on developing robust pathology and multimodal foundation models as principal machine learning scientist at Diagnostics, a leading European scale-up in AI-powered pathology, where he works to advance computational diagnostics for drug discovery and precision medicine. And the title of Dr. de Jong's talk today is Quantifying Foundation Model Robustness Against Medical Center Variation, the Robustness Index. So with that, Edwin, when you're ready to start, please feel free.
Thank you, Adam, for this kind introduction. So today's talk will be about measuring the degree to which foundation models reflect medical centers. And we argue that it's undesirable for them to do so, and we'll clarify why. This line of research started as 2 separate lines. I was working together with Eric and Jonas from the NKI at the bottom left. And in parallel, well, we didn't know it yet, at Diagnostics and TU Berlin, a team was working on batch effects, very related research. Meanwhile, I'm working at Diagnostics, and we joined forces, and this is now a joint research program, of which I'll share some first results. I'll briefly introduce medical foundation models and self-supervised learning, which should be familiar to probably a large part of the audience, and then introduce the main distinction of biological versus confounding features. And this is used as a basis to define robustness and also to measure it quantitatively. Now, as you're probably aware, medical AI is increasingly based on foundation models. So it becomes important to see how do these foundation models behave, what do they learn, and what is the effect when you use them as a basis for a downstream task. The setup is generally as follows. You have a patient from which you take a sample, which can be anything like an X-ray image or MRI or a slice of tissue that's put under a microscope to get pathological information, or nowadays even DNA, RNA, spatial transcriptomics, and so on. It doesn't matter too much which modality it is, or maybe even it's a multimodal model. Whatever the input is, can be an image or different representation, it goes into the foundation model, and what comes out is an embedding vector, just a long list of numbers. For example, let's say it's 1,000 dimensions, which is quite typical. And then this embedding vector is used as the input for downstream tasks. And the idea is that by having generalist medical foundation models, which have seen a lot of data, very diverse data also, it's possible to address many different downstream tasks. These models are typically trained using self-supervised learning. And at a high level, what these methods do is that they place similar samples closer together. So if you have 2 images from the same type of cancer, they will be drawn closer together in the embedding space and move dissimilar samples further apart. And the result of this is that these models learn to detect any differences between samples. So they can start to differentiate between different organs in the body, different cancer types, but also different visual characteristics, like the shade of color if you have a different staining in a pathology image. Now, we believe it's useful to distinguish between biological and confounding differences. And this is central, so it's important to get this clear. So with biological differences, we mean anything that's really representative about the true medical or biological state of the patient. So this could be the tissue type of the sample, the morphology, the cancer type, subtype, grade, and stage, any information really that's truly saying something about this particular patient. Any other information we will call confounding, because other information is potentially confounding. So when you see other differences, you might attribute the target that you're trying to learn in a downstream task to these differences, and that's almost always undesirable because you then learn the wrong correlations, spurious correlations also called. These confounding features, they do not give a useful contribution to downstream tasks because they are not really about the core thing that you're trying to learn. And by the virtue, or not virtue, but as a result of having correlations with the target that you're trying to learn, you can get bias in these downstream models, and that can harm performance of the predictions. Now, based on this distinction, we say that robust models capture biological features, and they are insensitive to confounding features. So it's a very simple distinction, but we found that it works in practice. And you can make this a little bit more technical by saying that a robust model has embeddings that correlate with the true biological features, and these embeddings do not correlate with the confounding features. Now, let's look at what this looks like in practice. So, as one of the earliest examples that I looked into, we trained a simple vision transformer model on TCGA, and then you get out these, in this case, 384-dimensional embedding vectors, which you can downscale, project to a 2D space, for example, using T-SNE or using UMAP. Other methods are possible as well. Now, I should add immediately that, of course, this projection only shows you a 2D view, so there may be a lot more information encoded, and there will be actually, in other dimensions. So you cannot draw too much conclusions from these images directly, but they can help actually to see, what is the model learning at a very high level overall. Now, one thing that's important to realize is that this projection procedure from, let's say, 1,000 dimensions to 2 dimensions is unsupervised. So it does not make any use of the cancer type, the organ, or even the medical center. No meta information about the images is used. And what you get out here is a scatter plot where each dot represents a sample, so represents a 1,000-dimensional vector. And if you look closely, you can see there's some clusters here, like in the middle you have this one, and you have a small one here at the top. Now, this makes us curious, what is represented in these clusters? And to get an idea of that, we can color these embeddings using meta information that we do have. So let's say we use the cancer type to color these embeddings. That's the first thing we looked at. Then you get this result. So notice that these are exactly the same dots. I'll switch back. You see it's the exact same location, but now colored with cancer type. And this at first sight looks very good. You see very nice, strong clustering. So all the red dots are together, all the orange dots are together, and so on. It's not true for all of them actually. So for light blue or cyan here, you see 3 clusters. So it doesn't always happen. And if you look closer, then you may see that, for example, in the glioblastoma cluster, which is a form of brain cancer, you do see 2 subclusters, this one here and this one at the top right. And you might wonder, well, why is there this subclustering? Why is it not just solid single cluster with a center and all dots around it? Now, the second thing we did is to color, again, these same dots, but now using the medical center that the data came from, as this is also available for TCGA. And then you get this picture. Now, to be honest, when I saw this for the first time, I was shocked. When I see this coloring, it's so blatantly obvious that the medical center is strongly represented in these foundational embeddings. That seemed to me a showstopper. It seems to me this is something you must solve if you're going to apply these foundation models in practice, especially in medical practice, where accuracy and bias are very important to be careful about. And so what you see here is that on the right, part of the glioblastoma cluster, the top right, is from Duke University, and another part, here a bit lower, is from Henry Ford. So even though the model puts all the glioblastoma patches in one corner of the embedding space, at least in this 2D projection, it does still make very strong separation between the two different medical centers that the data came from. And yeah, that carries with it a big risk. Suppose, for the line of argument, that you would just use these 2D t-SNE coordinates as the input for your downstream task. Well, in that extreme case, it's very clear that the location of the dot, whether it's in this Henry Ford area or the Duke area, will look to the downstream training procedure very different because they're in different locations. And that is undesirable and very risky. Now, in reality, of course, we don't train on these 2D t-SNE embeddings, but let's look at one more example. Here we see, again, a separation into medical classes, tumor versus non-tumor. And the orange and red dots are tumor patches, and the light and dark blue ones are normal patches. Now, on the left, you see an earlier foundation model, C-transpath. And what you see is that for two different hospitals, UMCU and RUMC, the orange and the red clusters are very far apart. So while these both represent tumor patches, they're represented in completely different parts of the embedding space. And in between, you have these two blue clusters, which are again for these same two hospitals, but representing the normal patches. And in this 2D projection, you see that a lot of the red tumor patches are on top of the blue area. And there might be an additional dimension where this is not the case, but at least in the 2D projection, you see this. So this could indicate a problem. Now, on the right, we see a more modern foundation model, UNI, trained with attention to diversity. And here we see that the orange and red clusters are very nicely grouped together, as you would like to see. And also in the blue area, you see only one orange triangle, whereas here for the earlier C-transpath model, you see a lot of them. All clear so far? Feel free to ask questions if something is not clear. So in an earlier part of this research that I did with Eric and Jonas, as mentioned, we took TCGA and then looked for a particular combination of biological classes, which are TCGA projects, which you see here in the columns of this matrix, so breast cancer, colorectal, adenocarcinoma, and so on. And medical centers, Astrand, GPCC, and so on. And we chose these not randomly, but in such a way that for all the 25 combinations in this table, a lot of them have data. And that makes it possible to construct a balanced data set. And for this balanced data set, we looked at how frequently, if you look at the k nearest neighbors in the embedding space, how frequently are these nearest neighbors representing the same cancer type in blue, and how frequently do they represent the same medical center in orange. Now, for these 5 models, except the bottom right, you see that for the first few hundred neighbors, the orange line is always above the blue one, right? Here, orange is above blue. Here it's the case, here as well. It's true for all of them. So for all these models, the medical center occurs more often in the nearest neighbors than the same cancer type. The only model for which this was not the case was Virchow-2. This is a model trained on the largest training dataset so far, millions of whole slide images. And here you see that for the first at least 100 neighbors, the biological class cancer type is more strongly represented than medical center, as you would like to see. Now, can you get this orange line to 0? In this plot, you cannot, because there will also be patches that both have the same cancer type and the same medical center. And in this result, in this earlier preprint, this was the result that you get. So you see Virchow-2 is a lot more robust than all the other models. But in follow-up work, we improved the metric to take care of that aspect that I just mentioned, that in some cases it's inevitable that you see some same-center patches. So when you consider which combinations can occur, so you consider, is the sample of a neighbor, is it from the same biological class, yes or no? So same cancer type, yes or no? That's what you see, that determines the row here. And you also check, is it from the same medical center, so the same confounding class, and that's the column here. Now, if a neighbor is from the same cancer type and the same medical center, so same, SS, we exclude that category from the analysis because it does not tell you anything about robustness. It might have placed this neighbor near because it has the same cancer type, but it might also have placed it near because it happens to be from the same medical center. And the same is true for OO. So when it's a different cancer type and a different medical center, you also don't know whether that's due to being of a different cancer type or being from a different medical center, so looking different visually. So the two remaining relevant combinations are SO at the top right, same cancer type, other medical center, and the converse here, OS. So it took me some time to wrap my head around this. So this categorization is about a relation always between two patches or two samples. But once you get to grips with it, it is a very simple concept. And it turns out that these fractions can be easily measured and are very stable across the embedding space. And let's have a look at that. So here, first let's look at the left, and you see two models. In the dark colors, so dark blue, dark orange, you see a modern foundation model, Conch version 1.5, which is a VLM, a vision language model, also trained on medical reports. And in the light color, you see an earlier model, C-Transpath. Now, as mentioned before, this SS class, where both the cancer type and the medical center are the same, that's the two blue lines. We're excluding that. But what you can see, and that's interesting to see, here you see information about a particular value of K. So at 300 here in the middle, this is information about, for a given reference patch, there's only a single neighbor at distance 300. And another patch in the dataset will have another patch, another neighbor at distance 300. But there's a very limited number of samples of neighbors that are at distance 300. So even though every K value is in a sense independent, these frequencies are developing remarkably consistently across the space. So this line could have gone up and down in very random ways, but that's not the case. So these foundation models seem to learn in such a way that they move the embeddings across the space, such that this information is really showing a consistent picture. It's a bit complicated way of saying it, but I think you see what I mean. Now, as mentioned, blue we exclude, same OO, so red we also exclude. So let's focus instead on the orange and the green lines. And if you have a robust model, you want this orange line to be high and the green line to be low for the nearest neighbor. So let's say the first quarter or so of this range of neighbors. Now, immediately you see a very clear difference between the modern Conch foundation model in dark orange here and the earlier C-Transpath model in light orange. There's a really big difference between these two lines, big gap. And in fact, when at the start there's this gap and it grows a little larger as you increase K to about 100, for example, and then it becomes smaller again. And after 300, it drops again. And this can be explained, but that's too detailed for now. So in green, we see the converse information. And here we see that the modern foundation model is able to avoid having these same medical center neighbors in its neighborhoods much more often than the earlier C-Transpath model. So both the orange line is higher and the green line is lower. So those are both representing a more robust model. Now, what KNN classifiers do is that they don't only look at neighbor K, they look at all neighbors from 1 up to K. And so we can aggregate the same information that you see here, and then you get this plot in the middle. And that makes the lines a lot more smooth and therefore even more stable, even though they were already very stable here. Now, the last step in calculating the robustness index is to normalize. So since we focus on only the orange and green line, we can normalize such that these always sum together to 1. So the dark orange line and the dark green line will sum to 1 in this picture. And that's why they move symmetrically here. And what you then see is that by this normalization, and by looking at this orange and green curve, we see that the modern conch model, indicated here at the top, has much higher values than the earlier Z-transpath model for this, for the relation between SO and OS, basically. So that's the relative frequency. And this is the quantity that we call the robustness index. So it's the normalized SO frequency, normalizing over SO and OS. Now, this is a bit abstract still. A little bit clearer maybe with the diagram. Here you see a reference patch represented at the middle. You see all its neighbors, or at least some of its neighbors. Only a subset of those are in these two categories of SO at the top right and OS at the bottom left. And that's the subset that you consider. And then for increasing distances here along this arrow, you consider which neighbors have the same biological class and which have the same medical center. And you want to have many of the ones on top and few of the ones at the bottom. And the robustness index is simply the relative size of the SO set compared to this total. That's all. Now, to evaluate this metric, we curated 4 multi-center balanced datasets based on existing datasets. All have multiple, so 2 or more biological classes and 2 or more medical centers. And they're based on existing datasets, like the Comedian dataset, TCGA, this is similar to the earlier paper, and Tolkar, a dataset on esophageal cancer. Now for TCGA, as I was explaining earlier, if you want to have a completely filled matrix with all the combinations, that's a very limiting, constraining criterion, which is a pity because there's a lot of data in TCGA and now we are only using a very small part of it. And to address that, there's a very simple thing you can do, which is just to look at pairs of cancer types and medical centers. So now we just, for example, look at BRCA, breast cancer, and colorectal, only those 2, and only Asterant and Christiana. So that gives you the top left 4 squares here. There's one combination that you can make. And for all of them, we use an equal amount of patches. So that's balanced. And this trick you can do 94 times with TCGA. So that gives you a lot of diversity covering many of the cancer types, many of the medical centers. And because you can average over these 94, or actually aggregate over these 94 combinations, you get a lot of data to base calculation on, which makes the result very stable. Here's some results. So here we evaluated 20 current foundation models. These are results for Comedian 16. You may wonder why the dot here is at a particular K value. Well, if you want to compare a single number, then you have to select one or more robustness index values from these curves. And the way we did that, which we think corresponds to at least one practical potential use case, suppose you use a KNN model to predict the biological class. Well, then you would typically choose a value of K that gives you good predictions. So for each model, we first optimized the K value that gives you the best biological prediction performance. And this gives you a distribution of different K values. And from this set, we took the median, with the idea that that's representative of these different models. So for Comedian 16, for example, we use a particular number of neighbors, I believe off the top of my head is 11, and look at the robustness index curve of all these different models at that particular K value. And that is the single scalar metric that we compare.
Hi, Edwin, do you mind if I ask you a question from the room? Thank you.
Yeah,
Go on, Mr.
Hi, Ben. Just a quick question from previous slides, please. The last one, yes. So I understand, like, for Comedian data set, then you say, like, you have 5,100 this is the images. But for TCGA, when you say 360, are these cases or image patches?
These are patches.
There are patches, okay. And how do you select patches?
In the first paper that's described in the archive preprint, we took, so that's this one, we took per combination, I believe, yeah, I have to check it, but a fixed number of whole slide images, and then from each whole slide image, a fixed number of patches. I believe it was 10 per whole slide image.
Yeah, makes sense. And do you have like a criteria to where to choose the patches from in each slide? Is it just random?
Yeah, so that's a difference between these two pieces of work. So here in this earlier work, it was a random foreground patch, and then with manual curation to make sure that the patches are informative and do not contain anomalies. But there are, limitations with this data set because it may not be informative. So the patch may not be informative about the tissue type. So it could be that it's that you cannot, no model could predict the cancer type from this particular patch. That could happen. So the biological task is relatively hard, and that's why the robustness index values are low. And to make sure that we use a sound selection of patches in this follow-up work, we paid special care to construct these data sets. And for example, for TCGA, it's based on TCGA uniform, which was already previously curated to contain balanced selections.
Thank you very much.
Okay, thank you, Edwin.
We discussed these results for Chameleon 16. We tried this on multiple data sets, as mentioned here, TCGA uniform. What's also interesting is that you see a quite similar shape of these curves for the different models. So, for example, here in blue, you see uni 2H, and it's first quickly increasing and then more slowly increasing. And for example, here, Prof Gigapath has very much the same shape. There's some variations between models, but overall, they have a similar pattern of how they distribute their patches across the embedding space. It varies a bit per dataset. That has to do with the dataset construction. This is the paired dataset that I showed. So 94 combinations, where for each of the 4 combinations you have 300 patches. And note that the SS and OO patches are excluded, so that leaves only 600 that are used actually in the robustness index calculation. Because we are including all neighbors, so the subset of this 1200 neighbors, when you use all 1200 patches, then all 300 SO patches and all 300 OS patches will be included because there's just no other patches. So necessarily all models will have a robustness index of 0.5 at k equals 1200 for this dataset. So that's a nice confirmation that makes perfect sense and that you would expect. So they all start somewhere and must end at this point. And the difference between these models is in how robust are they, how well are they able to place the biologically comparable patches near each other. And what is also positive to see is that there's a large spread of these different models. So at the bottom, you see very old models like SIGA, with a very low robustness index here for TCGA. And then it gets to more and more modern models. So at the top, you see here Conch version 1.5, version 2, and another version, the first version of Conch. And then Atlas, a foundation model that we train ourselves at AI Agnostics. Then last dataset, Tolkach esophageal cancer. Again, gives an additional data point. What's important is maybe not so much these particular results or these particular data sets, but it's also the whole methodology. This metric is very general. The only thing you need is embedding factors and a biological and confounding categorization. And this you can make for almost any foundation model in any modality. So even in proteomics, if you have a foundation model, you could use exactly the same metric. Now, to summarize the results, we also average over these 3 data sets. And then from each dataset, we have a median K value. Of these, we take the average. That's 24 here. And so this way we can make a comparison of all models. Now, it provides a stable metric, and it provides a very clear separation of the different models. So some of them have very low robustness index, and some of them a very high one. And now the question is, well, one first interesting question is that we looked at is, how does it correlate with dataset size that the foundation model was trained on? And in blue, you see the image-only foundation models. Now, there's a clear positive trend, but what's especially striking is that the models that are not only trained on images, in particular, for example, conch, both models, indicated in orange here, are way above this trend line. And to me, that's also a promising result. The way I interpret it, or we interpret it, is that the additional biological signal that you get from these medical reports helps the model or the training to identify, to find a more robust foundation model, even though the number of whole slide images is a lot smaller, multiple orders of magnitude, compared to other foundation models here at the right. Now you might wonder, what does this robustness index tell us? Doesn't really have an effect in practice. Well, to evaluate that, let's dive into the work that was done on the T. Lin side of this collaboration. What we see here is different samplings of training datasets. So on the left, you see the standard training dataset that's very balanced. But then you can increasingly correlate the biological and confounding class by making a certain selection of patches. So you could choose the patches in the extreme case at the right here in such a way that one cancer type is always from the same medical center. So cancer type 1 is always from medical center 1. Cancer type 2 here is always from medical center 2. Now, when I first saw this, I must confess, this seemed to me rather artificial. And of course, it is an artificial example. But it also seemed to me that this is hopeless, that if the downstream dataset has this balancing, it seemed to me at first there's no way that a model could give correct predictions. But the fascinating realization to me was that if you have a robust foundation model, you can actually get, you will get perfect prediction in this downstream task, while only with an unrobust foundation model, this is a problem. And that's because the robust foundation model only represents the true medical features, and so it will ignore this medical dataset, medical center, and an unrobust one has features for both the cancer type and the medical center, and so then the downstream training does not know which feature to use. So it will be confounded by these correlations. Because in the test dataset, yeah, your correlation might be different than in this training dataset. In test here is uniform, so we did not bias these test datasets, only the training one. I see a remark in the chat. I don't know if there was a question or I don't see the chat itself. But...
Hi, Edwin. Yeah, sorry, that was we had a question from David in the chat, who was just asking about the embedding space. I was wondering whether we could say whether we should say that to the end.
Sure. All right. So this is the example where you we constructed where you have an increase in correlation between medical center and cancer type, and we can make a very concrete and simple example of a downstream task to show why this is a problem for unrobust foundation models and not a problem for perfectly robust ones. So let's say that the dataset here with the perfect correlation, this will be an example of it. So you have just 4 normal patches in this very simple example and 4 tumor patches. And the normal ones are all from center A and the tumor ones from center B. Now, unfortunately, in your test dataset, this correlation is not present. In fact, it's opposite. So if the downstream model would use this medical center information to generate predictions, as happens actually in practice, then it would give exactly the wrong predictions for this test set. Now, the realization here is that if you have a robust foundation model, it will have features that represent the tumor type. So like this feature 1 here, but it will not have features that correspond to the medical center. So its second feature might be, for example, completely random. I'm showing here 0.5, but it could just be randomly distributed. or had just no correlation, so random zeros and ones. And so when a downstream training process has embeddings from this first FM1, it will very quickly see that even on this very small training data set, there's a very strong correlation between its feature 1 and the target, normal versus tumor, that's trying to learn. So it will use this feature 1, and as a consequence, it will make perfect predictions for the test set. Whereas a unrobust foundation model might also choose the same feature 1, but you would have to be very lucky because the features it receives are identical for feature 1 and feature 2. So it might just as well use feature 2, in which case it will give you exactly the wrong predictions. Now this is of course an extreme example, but it shows very clearly an effect that does happen to some extent in practice. So the issue is that these, when you have in an unrobust foundation model both these correlations, both features that represent biological features and features that represent confounding aspects, then these features can be entangled because there's no way for the downstream training to tell them apart because you have no further information. You are not using the patch, you are using directly these embeddings as input, so you lose other information that could help you solve this. And on top of that, these downstream datasets, they are typically small, at least a lot smaller orders of magnitude than the pre-trained dataset, and are also often in practice unbalanced because you might have different distributions, different populations for different medical centers. Now, when you do this increasing correlation, when you construct that, what is the effect on performance? So here we see from split 1 to 8 increasing correlation. And we see that for all foundation models, when you have a very strong correlation, performance will decrease. For the robust foundation models, the performance decreases less. So you see here, for example, at the top, a line that drops only a little bit, and here at the bottom, lines that drop a lot. And now the question is, of course, this performance drop, is it in any way related to robustness and maybe also to the robustness index? Well, it happens to be, especially for in-distribution data sets. So when you when you look at medical centers that are also in your downstream training set, there's a very strong correlation between this robustness index and the performance drop. So even though this robustness index is very simple to compute and directly based on the embedding space, you don't even need a downstream task to evaluate it. Still, it gives you predictive information about performance drops in downstream tasks. So this is, to us, an important result that draws the connection. It's not just some metric that might indicate something. It really does have a correlation with performance drops that you see when you train downstream models, also using linear regression. Because a counter-argument, people sometimes say, if I use linear regression, the downstream model will use the features that are capturing biological information. But that is not the case. We here clearly see that even with linear regression, these robustness issues are a problem that degrade performance. Now here even more concretely, in this chameleon example, H optimus 0 had a relatively low robustness score compared to these other models. And we looked at, so that means it makes, we also saw that it makes more prediction errors for chameleon. And we then looked at particular examples where it makes prediction errors. Now, at the top, you see a very clear tumor. This is the ground truth. And here, both Virchow2 and Conch very clearly find it, but not H optimus 0. And here, there's a more subtle one, with a small cancer area here, and even more tiny one here. That's probably hard to see. And here we see that the Conch model does find this tumor, which is impressive, whereas even a modern model like Virchow2 does not find it, and same for H optimus 0. So these robustness issues lead to incorrect predictions in practice on real images that may affect real patients. And we believe it's therefore essential to work on improving robustness, and that starts with measuring robustness. So in summary, we view robustness as prioritizing biological over confounding features. We saw that unrobust foundation models can produce entangled embeddings, where you no longer know which features correlate with biological aspects and which ones with confounding characteristics. This leads to prediction errors in downstream tasks, and even the best current models still make robustness-related errors. We devised the PathoRob benchmark, which consists of 4 balanced datasets, and introduced the Robustness Index. As mentioned, this can be used for all medical foundation models, not just pathology. It's a simple scalar metric. It's easy and fast to compute. You can just run a single KNN training and get the neighbors on the full set, and then that gives you all you need. It adds a second objective to foundation model evaluation, and I did not go into that, but so far foundation models are almost always only based on prediction performance. And this really shows a whole second dimension that we must start to take into account in evaluation of foundation models. Robustness is a different aspect, a different objective that we also must start to measure and optimize. And you may also imagine that in some cases, if you have 2 model candidates, one might be more having a better prediction performance, but the other one might be more robust. And actually in medical practice, robustness is essential because you don't want patients to be influenced by which hospital they happen to go to or which scanner they happen to be imaged with. So we view robustness as a second objective, and we believe the Robustness Index captures fundamental aspects of the embedding space, as we saw from these consistent lines. And with this metric, if you are starting to work on making your foundation model more robust, then you can easily use this metric to view the effect, to track the effects. To end, this dataset, so both the dataset, the benchmark, both the datasets and the code will be made available online. We're preparing a journal article on this. It's about to be submitted. And on coming Saturday in the morning at the ECDP in Barcelona, my colleague Julius from the TU Berlin will be presenting this in an oral. That's it. Thank you for your attention.
Well, thank you very much, Edwin, for a really fantastic talk. This is a really great piece of work. I mean, we're seeing so many new foundation model papers come out on a weekly, well, on a daily basis almost. So it's great to see that, well, that are often just percentages increase in our ROC. So it's great to see an actual paper that goes into actually looking at how robust these models are. So yeah, with that, we'll open the floor to questions. Does anyone online or in person have any questions they'd like to ask? I know there's a couple in the chat. I know David had one to start with, I think so. David asked, everything depends on embedding space, embedding metric, and embedding vector. Could you please say more about how these are defined? So I think you said you mostly used t-SNE, sorry. But do you want to go into more detail about that?
No, that's a misunderstanding. So t-SNE I only used for the visualizations.
At the start, okay.
It gave us a first impression that showed us that this issue exists. The whole analysis with the Robustness Index is based on the full embedding vectors. So on these 1,000-dimensional vectors. And this may sound like it's computationally complex, but KNN is very efficiently implemented, and it's actually very quick to get the nearest neighbors. So you can choose different ways to generate embedding vectors. In the first archive preprint that I showed, we used only the class token. In this work, we also take the patch tokens and the average of that and concatenate that, so you get a vector twice the length. And then for pre-processing, it's important to mention, as mentioned in the paper, we normalize using L2 so that all vectors have the same length. And when you do this, then the Euclidean distance is directly corresponds to the cosine distance. So then it doesn't matter whether you choose Euclidean or cosine, both give you the same ranking.
Sure. Thank you, Edwin, for going into a bit more detail there. I think we've also got a message from Pablo online who said, any idea on how to measure robustness if it is not feasible to have a biological label? So.
You might choose a different data set where you do have labels.
Thank you. So do we have any more questions from in person or online then?
Let's go with this.
Yeah, of course. No, you go for it first, Mr.
So first of all, Edwin, thank you. Can you hear me?
Yes, I hear you fine, thanks.
Thank you very much for really interesting talk. I really enjoyed it because I hardly like this whole topic and area of work. So just had a kind of silly question related to last slides. You mentioned that this can be a second dimension in which models can be evaluated. So was wondering, you might have point to it in your talk, but there are a few studies coming out and benchmarking these foundation models on prediction tasks, right? I wonder if you could have looked into the rankings that they present on prediction tasks, the ranking that you achieve, to see if there is a concordance between the algorithms that perform well on unseen data sets, and also in terms of robustness measures that you have.
Yes, good question. That's indeed would be a useful thing to evaluate. Yeah, I think that's a good idea. I think you also asked about the relation with performance, and for that, the graph that I showed with the model size is, I think, informative. So here we see that there's a positive correlation with dataset size, and we also know there's a positive correlation of dataset size with prediction performance. We also have plots that's not shown here, but in the article that we are submitting, we also plot prediction performance against robustness, so that will be useful to check. What I wanted to show here is that there are different objectives, different dimensions, and conch is a good example here. So, well, it does not directly show that, but you could see that there might be cases where there might be an overall correspondence between performance and robustness, but still you might have different model variants that have similar performance but different robustness, and we did actually see that. So when you vary the value of k, prediction performance varies and robustness varies as well, and that gives you many different combinations. Each k value is a single model that you can use if you want to create a KNN model, and by choosing k, you are choosing a certain trade-off between prediction performance and robustness. And that's only an example in the context of KNN, but this may be true in general. So if you start to make your model more robust, you may find one that's a lot more robust and a tiny bit lower in performance. And actually, the H0 mini model is the perfect example of that. In this plot, is not doing better, but in other plots like here, we see that for low numbers of neighbors, H0 mini, which is a distilled version of H optimus 0, has a much better robustness. And that's fascinating because it's a smaller model, distilled from the larger one, and it has similar performance, a tiny bit lower, but a lot more robust. So this may be actually preferable to apply in practice.
That makes sense. Thank you.
Thank you. I think we also have another question online from Clemens, saying that different foundation models output embeddings of different dimensions. Did you look at the impact of embedding size? I can imagine a less robust but more complex model could still be more desirable, as it would convey more biological information along with more confounding information that could be dealt with during training by increasing the dataset diversity. So did you get the main question from that? Sorry.
Yeah.
Yes.
So the question about the relation with model
size. Yeah.
Good question, and we did consider that. So here, the size of the dots, I did not go into that, but it represents the size of the model. And yeah, the example I gave earlier of H0 mini, it's a tiny model, but it has very good performance, and it's trained on a lot of WSIs. So that's why at this location. So there's no direct correspondence. It's not the case that a bigger model is always more robust, or the opposite. You do see a trend, but there may be confounders, right? Because the earlier models are smaller and they perform less. But yeah, H0 mini is a clear counterexample to that.
Sure, thank you. I think we've got a follow-up question from Pablo online, who said, is there any rationale between looking at the closest neighbors at the instance instead of at the slide level?
So that's also a good question. If you look at the closest neighbors, if you consider all neighbors, then the closest neighbors will very often be from the same cancer type and the same medical center. However, since we always look at only neighbors that are from SO, so other medical center, or OS, other cancer type, by definition, the neighbor is always from a different category and from a different slide than the reference sample.
Sure, that makes sense. Sure, thank you. Okay, then, well, does anyone else online or in person have any questions they'd like to ask Edwin? And Edwin, will you be at ECDP on Saturday then, or on Thursday even?
I myself will unfortunately be not, but my colleagues will be there from.
Okay.
Yeah.
Sure. I think there's quite a large contingent from our centre going. So.
Oh, great.
But yeah, does anyone have
a quick question?
Oh, sorry, Mark.
So did you see any evidence that the kind of the center info in the embedding space was concentrated in maybe a small subset of the features? And do you think there's any possibility of like seeing if we can find the features which tend to encode the center information and then remove, kind of ignore those features, and then how kind of an embedding space which concentrates on the biological information that way? If that made sense.
Yeah, that makes perfect sense. So you're asking whether you could see which embedding dimensions correspond to cancer type and which correspond to medical center.
Yeah.
Could even generalize that a bit further. There have been lines of research in LLM research where they use sparse autoencoders to take the embeddings and expand them to a higher dimensional space with very sparse activations, with the hope that you get an even more clear separation between these different concepts. There have also been recent reports that the success with this approach is limited. It's definitely something you can do and would be interesting to analyze. We have not analyzed it in this paper so far.
Yes.
Sure. Thank you. And I suppose, yeah, one final question for me then is, so we're halfway through the year now. What do you expect to be in terms of computational pathology? What do you expect to see over the next 6 months in terms of advancements? We've obviously seen lots of foundation models over the last 2 years now. So yeah.
Yeah, I expect and hope to see more and more robust foundation models. Now that there's increasing awareness, hopefully also with this talk, of the issue of robustness, I think it's important that everyone in the field starts to pay attention to it and starts to analyze it, whether it's with this metric or other metrics. Because I really believe that it matters for the patient. This line of research started when I worked at ScreenPoint 7 years ago in radiology, breast cancer AI. And we noticed that for hologic scanners and Siemens scanners, the models that we trained gave different results. And yeah, already back then, it was very clear to everyone in the company that that's undesirable. But what's concerning to me and others and everyone in our team here that it is work is that this same issue still plays a role in current foundation models, and maybe actually even more so, because when you think about what self-supervised learning does, it tries to learn all differences. So if you don't tell it which are the biological differences that you care about, it might actually learn both equally well and give you a very unhelpful representation to train your downstream model on.
Thank you. And I think we have got time for an extra question. There's one online. So Azam has said, as a follow-up question, when examining the correlation between the performance and robustness, since you're searching among patches, do you distinguish between patches? Okay, I think we've already had that question, but it was okay. Do you distinguish between patches from the same side and those from different slides that originate from the same center? I think we had a very similar question already, but.
Yeah, so the neighbors will always be from a different slide because they either have a different medical center or a different cancer type.
Yeah, perfect. Yeah, sure.
So I was just going to ask, looking at this graph, whether there's any sort of metrics to diversity of data that you could include in this. So like Conch, I know, is included IHC and stuff, and Uni isn't. So is that another reason why it could be more robust?
That is absolutely an important aspect to look at, and it could well explain part of the differences. Yes, good point. Yeah, we know that some of these models up here are trained also on IHC data, and I could well imagine that we have not tested it, that including IHC data and in general any augmentations as already done in dyno trainings helps to somewhat improve the robustness. I think the challenge for the field is how to really maximize robustness and really focus on this to get the most robust models possible.
Well, thank you very much, Edwin. Thanks, George. So yeah, I suppose with that we'll draw the talk to a close. So so thanks again, Edwin, for a really interesting talk. And our next talk will be on Monday, the 7th of July, so 2 weeks away, the usual time of 2 till 3 p.m. We're going to be joined online by Dr. Rohan Chakraborty from UCL London, and hope to see you all there. But yeah, thanks again, Edwin, for a really, really great talk, and have a good rest of the day.
Thank you, Adam, for the nice invitation and all your attention.
See ya. Thanks, everyone.